Welcome to our second video on oxidative phosphorylation. In this video, we really want to take some time to look at how metabolites get across the mitochondrial membrane and the regulation of this cycle. All right, so just kind of a reminder that there is a whole lot going on in the mitochondria. We have the citric acid cycle. So this is our inner mitochondrial membrane. So out here we have the inner membrane space, the cytosol, and here we're inside the mitochondrial matrix. We have the citric acid cycle. We have the electron transport chain moving the electrons. We have the ATP synthase where we're converting ADP to ATP. And because there's so much going on, we have a lot of metabolites that we have to get across this membrane. But the inner membrane is not permeable to small molecules at all. Everything needs a transporter molecule. So this inner membrane is packed full of transporters that transport metabolites back and forth across this membrane. We're going to look at a couple of NADH shuttles, and we're going to look at how ADP and ATP get across. We're going to start by looking at NADH shuttles in the muscle cells. So this will be different in other types of cells, but we need a way for the NADH created in glycolysis in the cytoplasm, right? We need to be able to get those electrons from that cytoplasmic NADH into the electron transport chain. Okay, and the way we do this is what we call the glycerol phosphate shuttle. We're going to look at a picture. Okay, but in short, we're going to transfer those electrons. So those electrons that start with in our NADH are going to get transferred to FAD and then to Q. Okay, NADH is a huge molecule, and there just aren't transporters that'll take that whole molecule into the matrix. All right, let's take a look. Here's our glycerol 3-phosphate shuttle. So up here, we're on the cytoplasmic side, okay? Down here, we're in the matrix. You can see that labeled. So the NADH is going to pass its electrons. So here we are with our NADH to start. It's going to pass its electrons to this dihydroxyacetone phosphate. And if that sounds familiar, it's because we've seen it as a glycolytic intermediate. And in doing so, we're going to get the reduced form, this glycerol 3-phosphate. And that molecule now carries the protons and electrons from the NADH. They're now embedded in this glycerol 3-phosphate. And it's going to pass those off to the FAD, which is a prosthetic group with this mitochondrial enzyme. Okay, so we have this enzyme right here, mitochondrial glycerol 3-phosphate dehydrogenase. But notice that this is associated, right, with our inner membrane, and the FAD is a prosthetic group attached to that. That FAD is going to pass the electrons down here to Q, okay, to form our QH2. And from there, they're going to get carried into the electron transport chain. They get carried into complex 3 of the electron transport chain. So that's how we're getting those high energy electrons across the mitochondrial membrane for the muscle cells. And in the process, notice that it's important we're regenerating our NAD+ so that glycolysis can continue. Now in the heart and liver cells, this is a more complicated process. So in the heart and liver, electrons from cytoplasmic NADH are used to generate mitochondrial NADH. So ones made out in the cytoplasm are used to generate mitochondrial NADH, ones in the cytoplasm, through what we call the malate aspartate shuttle. So what you're looking at over on the right here was the one we just looked at, okay, the glycerol 3-phosphate shuttle. And here is our malate aspartate shuttle that we're about to look at. Okay, notice that they both at the end work to get those electrons into the mitochondrial matrix. So when we look at this, okay, this is a complex system that has this is what I wanted to highlight for you real quick, right? 2 membrane transporters, 4 enzymes. You do not need to know the names for all these enzymes. I really want you to know the big idea of this. And the main point is that we have different systems to get electrons through the mitochondrial membrane depending on the cell type and organism. But we need to be able to get those electrons into the mitochondria, and we need to regenerate that NAD plus in the cytosol. So again, when we look up here, we've got our cytosol on this side. We've got our inner mitochondrial membrane here, right? And then the matrix. We have our high energy electrons as part of that NADH. And what we see is that the oxaloacetate picks up those electrons to form malate. Oxaloacetate can't move through this shuttle, but the malate can be carried into the matrix. And when it is, it's oxidized back to that oxaloacetate. So look what we've done, right? Up here at the top, we started with NADH, and we end up regenerating our NAD+. And at the bottom, we're taking some NAD+ that was already in the matrix, and we're generating NADH. So we are, by a seemingly complicated route, transferring those electrons. So now we have NADH inside the matrix. Now that's all well and good, but we need to be able to get this oxaloacetate back out, right? We've got to be able to do this whole thing again and again. All right? And so when we do that, we actually have a process that's called a transamination reaction, where this oxaloacetate gets converted to aspartate glutamate to the alpha keto glutarate. And each of these has a shuttle that will take them out. So notice that this shuttle on the left carries malate in and alpha keto glutarate out. So it's an antiporter, right? One going in, one going out. The other one is also an antiporter and allows for the exchange of glutamate coming in and aspartate going out. Okay, so we need to get the electrons, high energy electrons, into the matrix so they can be used in the electron transport chain. We also need to be able to get ADP into the mitochondrial matrix. The mitochondria is making ATP for the whole cell, but to do that, it needs the ADP, and we need to be able to get the ATP out to the rest of the cell. The enzyme that is used for this is the ATP-ADP translocase. Okay, it lets us exchange these. So look what we've got. We have ADP in the cytoplasm, ATP in the matrix. So we've made ATP in the matrix, and we've used some of it up, so we've generated ADP in the cytoplasm. We need to reverse those, and that's what this ATP-ADP translocase does. It takes that ADP into the matrix and the ATP out of the matrix. Okay, this is powered by that proton motive force. I want you to think back to when we did the what's the word I want? The membrane transport. When we did videos on membrane transport, and we talked about that, and we calculated the Gibbs free energy that included the chemical gradient portion and the electrical potential. This is what we're talking about, and we're going to have some practice problems to look at for that. All right. This is actually, among our various cartoon pictures, one of the simpler ones. If you start in the upper middle piece, you can see that we have our ADP, ATP translocase, so shown here, these two pieces, and it's open to the cytoplasm side. Well, there's a lot of ADP in the cytoplasm, so one comes in. So here's our ADP molecule. From that ADP coming into the site causes a conformation change where these things close up on the outside, open up on the inside. And so when that happens, that ADP will flow out. Okay, there's not much ADP here, right? It naturally wants to go into the area of lower concentration. This is now open. There's a lot of ATP, so one will flow in. When that happens, it causes a conformation change that closes this, opens it to the outside so the ATP can exit. So one enters, one exits, one enters, one exits, one and then the other. If we go back up here and we look, we notice and we remember that the ATP has more of a negative charge. This helps it be drawn to the cytoplasm side of the cell because the outside of the mitochondria in that inner membrane space has more positive charge. So the mitochondria is negative on the inside, positive on the outside. So that's another driving force for this ATP wanting to move that direction. All right, so mitochondrial transporters allow metabolite exchange, right, between cytoplasm and mitochondria, the way we can get things in and out of that inner mitochondrial membrane. One thing I want to point out here is that the ATP-ADP translocase, that one we just looked at, the phosphate carrier, and the ATP synthase are associated with one another to form a large complex, the ATP synthasome. We already looked at how ATP formed dimers and how that influenced the shape of that inner membrane. And we looked at, and then we know the electron transport pieces tended to be right close to there so that they could provide the electrons needed and the proton gradient. And what we're now seeing is that these things are all closely associated with each other, right? So for the ATP synthase to work, it's got to have ATP. So it makes sense that it's located close to this. It has to have phosphates, so it's located close to that. So these things all work together. So ATP is generated in the mitochondria, ported out of the mitochondria, and the overall complete oxidation yields 30 molecules of ATP. Most of those are from this oxidative phosphorylation, the pieces that we have been looking at now. So we're using the redox reactions to essentially power our addition of the phosphate onto the ADP. 26 of the 30 are from that. The remaining 4 are from our glycolysis piece. And if you remember, we got 2 ATP directly from that, from substrate level phosphorylation. And then we got NADH. If glucose undergoes fermentation, only 2 molecules of ATP are generated. And that's because if you remember in that fermentation process, the NADH, so these high energy electrons are used to make more ATP. Those were used up in the fermentation process to regenerate the NAD+. So we get a little bit less ATP. And this last part, only 2 molecules. So when glucose undergoes fermentation, only 2 molecules are generated. It's talking about from those 4. All right, rate of oxidative phosphorylation is determined by the need for ATP. That shouldn't be a surprise, right? So electrons are not going to flow through the electron transport chain unless this ADP is available. Okay, so the regulation of this entire oxidative phosphorylation process is called acceptor or respiratory control, and it is an example of that energy charge, right? That ATP to ADP or ATP to AMP ratio. If we have lots of ADP, if we have a high ADP concentration, we expect that the electron transport chain will run, ATP synthase will run, right? That's all controlled by the energy charge. Okay, following this, there are practice problems. This is the type of math we've already done earlier when we talked about membrane transport. So I will work these out in a later video, but I want you to try them on your own first. So I've put them in here. There are 4 problems. So give those a try, and we will work them out in a later video.